Namaste. Welcome to this module on animal ethics in biomedical research. I am Professor Mathangi from Chettinad Academy of Research and Education. As we are doing a course on research, I definitely wish to have this disclaimer that some of the images that I've used in this module have copyright, and I've used them here in this session only for better understanding. This module is divided into three parts. In the first part, we'll be seeing the importance of animals in biomedical research, the major discoveries that have come out using animals as experimentation model, what's the need for the use of animals in research, and the experimental animals predominantly used. In the second part, we'll be seeing the laws and regulations that govern the use of experimental animals and the Indian CPCSEA regulations that govern the use of experimental animals. In the third part, we'll be seeing the guidelines that has to be adhered to in the use of animals, the 3R principle and the alternatives to animal experimentation. Here we begin with the first. The use of animals in experimental research starts from as early as the development of medicine, which leads us to the ancient Greece. It starts from Alchem Cotton, the father of neuroscience in the 6th and the 5th century BC, Aristotle in the 4th century, Erastius and Heliophilus from the 4th and the 3rd century, and Galen in the 2nd and the 3rd century AD. I wish to quote here what William Harvey has said: "To learn and teach anatomy not from books but from dissection, not from tenets of philosophers but from the fabric of nature." The birth of experimental medicine actually started with Claude Bernard from the 19th century. He said, "A properly controlled and rigorously conducted animal experiments provide reliable information on physiology and pathology of medical relevance." He said that introduction of study of experimental medicine is the most important thing that is required for the development of the medical field. The importance of biomedical research and animals used in that. This actually begins right from understanding the basic physiology of every system. Today, we have a world without smallpox. Very rarely do we have people who are afflicted with polio. We do have cardiac surgeries, major surgeries, like cardiac surgeries or transplant, to as simple as cataract and as specialized as the microsurgery of hand, have all been possible because of trials that have been done in animals, mastered and then done on humans. The treatment that we see for diabetes or for cancer are basically from the experiments that have been conducted in a very rigorous manner in the animal studies. The various vaccines, antibiotics that we use are again because of the use of animals in the biomedical research. The drugs that we use, right from as simple as aspirin to the toothbrush that we use or the sunscreen that we use, are all the benefits that we have gained from use of animals in the experimental research. Not only human, even our pet animals, be it dog, cat or our farm animals, have all been treated of various debilitating diseases that would have afflicted them, and we would have lost our pets or our livestock to reap the benefit from the studies that have been conducted on the experimental animals. Let's go further to see the major breakthroughs that have been coming out from the research using experimental animals. As we saw, in 150 AD, we had Galen, who did the systematic study using various animal models. In the 1600, we had the Servetus Vesalius Harvey for the way circulatory system that has been explored completely, predominantly using dogs as the experimental model. We have the smallpox vaccine that has seen to it that this disease has gone off the globe by Edward Jenner using the animals that have been done, experiments done using cattle. Louis Pasteur's experiments, which were very systematic, done in the 1881 gave us the germ theory, the rabies vaccine, and many more that we are having the benefit of it today. Robert Ross studied the entire life cycle of the malarial parasite and helped in having a treatment for malaria, diabetes, which would have killed many people in the globe, has the treatment of insulin because of the number of dogs that have been used by Banting and Best. Polio would not only have debilitated the young children but even the adult. We have the vaccine today. Thanks to the experiments that have been conducted by Salk and Sabin, we have the polio vaccine now. The coronary bypass, which is the main surgery that we are having for the cardiac problems that we see today, have been perfected by the experiments that have been conducted in dogs. It's just not coronary bypass alone. Dogs have been the model for us for valve replacement, for heart transplant, and many more cardiac things that have been done today to save many more lives that we are having now. Not only this, we do have our first animal that went to the space, Laika, that helped us to know if human can be sent to space as well. All these things could be possible only because animals were used as the first trial model because before they were tried on the human. Now let's see, what are the animals that are predominantly used? The animal that are predominantly used starts from the mouse, the rat, guinea pig, rabbits, to name the small animals, to go further to the dogs and to the non-human primates which have been used. Apart from this, frogs, fishes, reptiles and birds have also been used for the various discoveries starting from diphtheria, polio, typhoid, measles, smallpox vaccine, the rabies vaccine, the tetanus vaccine, etc. Any development goes from the small animal to the larger animal to the non-human primates before we try them on the human. Now let's see the major discoveries that have come out of animal experimentation. Each of these discoveries have won a Nobel Prize or are worth the Nobel Prize in itself. In 1600, we had the circulatory system and the functions of the lungs that were deciphered using studies that were done on animal models. In 1700, Hales studied the measurement of blood pressure and things that could change the blood pressure. The vaccines and the immunity, how it develops, understanding the infections were done in the 1800s. In the 1900s, we learned what are antibodies, understood the hormone system. In 1930 onwards, we had the major breakthrough in the medical research using animals, like starting from the nerve conduction to the tumor viruses, understanding the embryonic development, how the muscle activity could be controlled, the energy metabolism, how we hear, to the development of monoclonal antibodies, the biochemical functions of the liver, understanding the transplantation antigens, the functions of the brain, prostaglandins, development of the transgenic animal models, understanding the basics of memory, autoimmune diseases, in vitro fertilization, and the most important discovery of the last century, cloning, gene manipulation, the cancer markers, etc. We had first sequenced the mouse genome and tissue engineered organs that have been developed which are worth transplantation. Animal studies have helped in developing the new drugs and vaccines like insulin for diabetes, various vitamins, anesthetics, diphtheria vaccine, the broad spectrum antibiotics, whooping cough vaccine, polio vaccine, antihypertensive drugs, rubella vaccine, drugs for ulcers, anti-asthmatic drugs, drugs for leukemia, drugs for leprosy, immunosuppressants, drugs to treat various viral diseases, and to the latest gene therapy as well. There are a number of surgeries and diagnostics that have got modified because of trials that have been done on the experimental animals, like the heart-lung machine, dialysis, renal transplant, cardiac valves, pacemaker, the hip replacement surgery, corneal transplant, coronary bypass, heart transplant, the improved methods of sutures and other surgical techniques, the CAT scan, the life support system for the premature babies, and the laparoscopic surgical techniques. And why are animals used? What makes them so special? It's because animals have very similar organ and organ system to the human. Their susceptibility to the various diseases are almost identical. And most importantly, we share common ancestors. That has got a similarity in our DNA. In fact, we share 99% of our DNA with the chimpanzees. With the mice, we have 98% of our DNA, which are very similar to that we see in the mice. And most important, if there is a disease, if there is an exposure to a toxin or an environmental change, does it affect this particular generation or the subsequent generation? We can't wait for generations of humans to see this change, but experimental animals helps us in this process because we can see what impact these debilitating conditions have for the whole generation of that animal as well as to the subsequent generation. Is it carried over through the generations? Can be done. What happens if the environment is being altered, like as simple as diet, temperature, or even light or noise? If they are altered, what happens? All these manipulations could be done on experimental animals. Hence, they become a suitable model to explore the changes that would impact a human in itself. Now, what are the animals that are predominantly used in our research and for what purpose are they being used? If we see this pie chart, it clearly shows experimental animals have been used not just for education purpose or toxicological studies or production of quality control or for R&D, but predominantly to explore the fundamental nature of our biological system. And this pie chart clearly shows the experimental animals which are being used. The most predominantly used one are the mouse. The next are the rats, subsequently followed by the guinea pigs, rabbits, fishes, birds, and other species. And the most important use of the animals have been for the drug research and testing of various vaccines and other drugs as well. This graph clearly shows the various animals that are being used. And again here we see the mouse and the guinea pigs which are predominantly used. This set of graph shows mice being predominantly used, and the use of the animal have been changing over the decades. Now, the model that is gaining more importance is the zebrafish model, apart from the rats, guinea pig, mice, monkeys, and the dogs. With this, we come to the end of part 1 of this animal ethics in biomedical research.